Hello and welcome to my channel. So my topic for today is hematopoiesis or hematopoiesis. So basically, what is hematopoiesis? It is the process of production of blood cells, which are the RBC, WBC and platelets. And where does it happen? In the bone marrow. So all the blood cells which are circulating in our body are produced by bone marrow. Now we come to the natural history of hematopoiesis. So basically it starts at 3 weeks of gestational life or fetal life in the yolk sac. So the first time the blood cells are produced in the yolk sac. Now at 3 months the responsibility is shifted to liver. So liver takes the responsibility of producing blood cells. Then at 9 months the responsibility is shifted to spleen, lymph nodes, then gonads, which are the male testes and female ovaries, and then the posterior abdominal wall. Now at birth, all bones start hematopoiesis. Then at puberty, long bones like for example the humerus, the femur, tibia, fibula, etc., and the vertebral column, they start hematopoiesis. As we all know that bone has an inner medulla and an outer cortex. So normally hematopoiesis takes place in medulla, which is called the medullary hematopoiesis. If medullary hematopoiesis stops after puberty, then hematopoiesis can start in liver, spleen, lymph node, gonads, and posterior abdominal wall. And this type of hematopoiesis is called the extramedullary hematopoiesis. So before we start the process of hematopoiesis, we must know that the conversion of one type of cell into another is under the effect of cytokines, having autocrine effect. Now, what are cytokines? Cytokines are proteins that carry a specific message or signal to the cells. Now, what is autocrine effect? So basically, when the cell secretes cytokines, these cytokines act on the same cell itself and differentiates them into different cells. This is called autocrine effect. So now we come to the process of hematopoiesis. The pluripotent stem cells, which are the precursor cells, and they have the ability to differentiate into different types of cells. These pluripotent stem cells are the cells from which all the blood cells are formed. The pluripotent stem cell differentiates into two types of cells: the lymphoid multipotent stem cell, also known as the lymphoid stem cell series, and the myeloid multipotent stem cell, also known as the myeloid stem cell series. So now first we come to the lymphoid multipotent stem cell. They convert into three types of cells: the early pre-B cell, the early thymocyte, and the early natural killer cell. These cells are the immature cells that are finally going to form the mature cells. So first, the early pre-B cell, which is an immature cell, is converted into pre-B cell, and then the pre-B cell is converted into B lymphocyte. The early thymocyte is converted into common thymocyte and then into the mature T lymphocyte, while the early natural killer cell is converted into mature natural killer cell. Now the myeloid multipotent stem cell is converted into three types of cell. The first one is the colony forming unit, GM. G means granulocyte and M means monocyte. And then the colony forming unit, E. E means eosinophil. Then the colony forming unit, EM. E means erythrocyte and M means megakaryocyte. The abbreviation of CFU is colony forming unit. So the colony forming unit, GM, differentiates into granulocyte series and the monocyte series. So first we come to the granulocyte series. The first immature cell that is formed is the myeloblast. It is converted into pre-myelocyte, then myelocyte, and then the metamyelocyte. The metamyelocyte is then converted into granulocyte. The granulocyte is differentiated into neutrophils and basophils. Neutrophils are the most abundant of all blood cells. Granulocytes include neutrophils, basophils, and also eosinophils. We must remember that granulocytes are the WBCs having granules in them, and granules contain enzyme, different types of enzymes. Agranulocytes include monocytes and lymphocytes. Now if somebody asks you what is the difference between granulocytes and agranulocytes, so one thing we must remember that agranulocytes also contain granules, but they don't get stained easily. That's why they are called agranulocytes. Basophils contain histamine granules. When they are located in the blood vessels, they are called basophils. And when they cross the blood vessels and go into the connective tissue, they are called mast cells. So in blood vessels, basophils. In connective tissues, mast cells. Now we come to the monocyte series. The first immature cell that is formed is the monoblast. The monoblast is converted into pre-monocyte, and the pre-monocyte is differentiated into the mature monocyte. In blood vessels, they are called the monocytes, and when they cross the blood vessels and go into the connective tissue, they are called macrophages. So in blood vessels, monocyte; in connective tissue, macrophages. Size-wise, they are the largest WBCs found in the body. Now we come to the colony forming unit, eosinophil, also known as the eosinophil series. So the first immature cell that is formed is the eosinoblast. Eosinoblast is converted into eosinocyte, and they finally mature into the eosinophils. At last we come to the colony forming unit, erythrocyte series and megakaryocyte series. So the first immature cell that is formed from the erythrocyte series is the erythroblast. The erythroblast is converted into early normoblast, which is basophilic. Then it is converted into intermediate normoblast, which is polychromatic. The intermediate normoblast is then converted into late normoblast, which is orthochromatic. The basophilic, polychromatic, and orthochromatic are because they get stained differently with different colors. The late normoblast is converted into reticulocyte, which is also called the baby RBC. And finally, the reticulocyte forms the proper mature RBC, which is also known as the erythrocyte. The first cell that is formed in the megakaryocyte series is the megakaryoblast. This megakaryoblast increases in size and matures into megakaryocyte. As the name indicates, megakaryocyte is a large cell. So what happens is that cracks appear in the megakaryocyte and it is broken down into smaller platelets. So from here we get proper mature platelets. And with this, the process of hematopoiesis in the bone marrow is completed. In the next video, we are going to talk about B and T lymphocytes, how they structurally and functionally mature, and how they perform their function. If you like this video, kindly like, comment, and subscribe my channel.